Hello, and welcome to this Nature.com custom webcast titled Cost-Effective Miniaturized Next-Generation Sequencing for Microbiome and Metagenomics. My name is Joshan Karpin, and I will be your moderator. Today's webcast is sponsored by Labcyte. We'll begin the webcast with presentations from Jefferson Lai and then Dr. Nur Hasan. We will then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. And now over to Jefferson.
Hello everybody, and thank you so much for joining us on the webinar today. I'm Jefferson Lai, application scientist at Labcyte, and today I'm going to be talking about cost-effective and miniaturized next-generation sequencing for microbiome and metagenomics applications. Before I get started, I wanted to say a thank you to Nature for hosting this webinar, as well as CosmosID and Dr. Hasan for collaborating with us, providing the bioinformatics tools necessary to analyze and visualize this data. And you'll hear more from him shortly after my talk. So, before we get started, I did want to introduce the Echo liquid handlers that Labcyte produces. Here on this slide, you'll see the two major models that are out there in the field, the Echo 525 and the Echo 555 or 550. So these are liquid handling instruments that do not use any tips. We transfer liquids using acoustic sound energy that is very low amplitude. So samples are transferred with high fidelity. You'll see that the Echo 525 produces 25 nanoliter increment droplets, and we can transfer 200 to 400 droplets per second, resulting in about 5 to 6 microliters. And with the 555 and 550, those are 2.5 nanoliter increments, transferring at a similar but faster rate to a total 500 to 600 nanoliters per second. And so these instruments differ mainly on that flow rate. Depending on your uses and applications, you may choose one over the other. So going to the next slide, we just wanted to quickly introduce that Labcyte this year has released some new instruments. They're the Echo 650 and 655, and they are very similar to the 550 and 555, but have been updated in various ways. We have new software, redesigned fluidics, redesigned transducer focus assemblies, and a plate dryer that has been redesigned. And so 650 and 655 are very comparable to 550, 555 in the terms of 2.5 nanoliter increments of transfer. And there is a path of upgrade available. So you'll see 650 can be upgraded to a 650T, and a 655 can be upgraded to a 655T. And the T really allows for users to use Brooks designed 96-well acoustic tubes. This way, users are not locked to a plate, but my individual tubes can be pulled depending on what is necessary for the experiment at hand. Here's a quick slide to go through the technology behind the Echo. So at the bottom, you'll see a transducer focus assembly, and this is where the transducer will come up and use coupling fluid to make contact with your source plate. We send a very soft ping into the well and listen for the echo, and based on the return of that signal, we are able to use our proprietary technology called dynamic fluid analysis to determine the viscosity of the liquid as well as the well height. So we know exactly how much liquid is in a well, as well as the viscosity and how much sound energy we then need to use to precisely eject our droplets. And you'll see on this slide that we cover solutions from glycerol 0 to 50%, buffers, aqueous, surfactants, crystallography reagents, and DMSO-based reagents. And so this has been applied in many different ways towards cell lysates, various reagents, glycerol storage solutions, DNA, RNA. There's a whole host of solutions that can be easily transferred with the Echo with very minimal user input. The instrument determines much of this on its own. Okay, so that's the intro, and now I'm going to go into miniaturizing next generation sequencing. So here we're asking why Miniaturized NGS? Because miniaturization allows for an expensive workflow to be run at an order of magnitude lower cost, thus broadening its uses. So by miniaturizing, we're reducing the reagent costs, reducing the amount of sample input required, as well as liberating human time. Here you'll see in this chart, we have a couple examples of full-size protocol as well as an Echo system enhanced protocol, which is for the most part miniaturization. Kappa Hi-Fi 2X Ready Mix is what was used in our 16S rRNA Amplicon sequencing workflow, and Illumina Nextera XT is what was used for our whole genome sequencing of microbiomes. And so you'll see that the Echo is able to reduce the reaction volume tenfold easily, and therefore we can really reduce the amount of cost of reagents per sample run. Now, these are based on a tenfold reduction in volume. 20-fold reduction in volumes are possible, but oftentimes the magnetic bead cleanup on the back end serves to be the limiting step. And so we want to make sure that miniaturization makes sense, and that when we get to the end and run to the bead steps, that it's still a volume that can be handled. Okay, so Amplicon sequencing of 16S rRNA regions has long been the standard technique used to assess patient microbial diversity. We can miniaturize these amplicons to use much less reagents, though the savings in these PCR mixes isn't as drastic as Nextera XT, for example. But there are various limitations in 16S rRNA sequencing. For example, the eukaryotic and viral components of the microbiome are missed. You can use ITS1 for fungal diversity, but the viral components are still missing. And this requires repeated experiments. And if you survey one region on the 16S rRNA, then that information is typically accurate only to the genus level. To reach species level, typically more than one variable region needs to be surveyed. And most importantly, strain-to-strain variation is what is responsible for pathogenicity, toxins and virulence factors, epitopes, antibiotic resistances, and various other characteristics that can make a commensal strain into a pathogenic strain. So really the functional unit for microbiome is the strain. That is the differentiating factor. Whole genome sequencing can determine the strain-to-strain variation. Whole genome sequencing is great in that sense because it covers the entire genome, and whole genome sequencing can also be used for metagenomic homolog discovery from uncharacterized organisms. This is oftentimes of interest to people looking for enzymes or pathways homologous to what we are studying but haven't really been discovered, or just exploring the diversity of what's out there. And so by miniaturizing whole genome sequencing, we're making the technique to be more cost effective and thus approaching the cost of running a 16S rRNA amplicon sequencing reaction, and in hopes of one day it could potentially replace 16S rRNA amplicon sequencing as a whole due to the much richer amount of information that's provided. So here on this slide, you'll just see that companies are leveraging miniaturization, and oftentimes the equation is based on how much miniaturization to make the process really cost effective versus how much volume is created on the back end. So here we'll see that a synthetic biology company, Amyris, has used the Echo to miniaturize their workflows tagmentation by 100-fold, making that very, very cost effective for sequencing of their plasmids. Okay, here I'll jump into the workflow for miniaturized 16S rRNA amplicon sequencing. We have our sample microbiome, which is then the gDNA from that is purified, and then we use PCR to amplify a variable region of interest. In our case, we use the V4 region of interest and thermocycled that 25 times. Then did a bead cleanup, and then we had a purified PCR product with alumina landing pads. Then for the second PCR, we took the product of that and performed a short cycle, just 8 cycles of PCR amplification to add on unique indexing primers, and then did a bead cleanup and purified that product, which was then put through library QC, so fragment analysis as well as quantitation. And then the Echo was extremely good at pooling all these samples together and normalizing simultaneously. So because the instrument can shoot at 25 nanoliter or 2.5 nanoliter increments, we're able to accurately eject the amount of total DNA wanted per sample so that we reach equimolar amounts. For these experiments, we focused on using the Echo 525. And then we denature the libraries and load them onto the sequencer. In our case, it was the Illumina MySeq. And then software analysis of the raw data was performed mostly in CosmosID. So here I'm just walking through some more technicals of the workflow. You'll see that in this Miniaturized 16S rRNA Amplicon sequencing, we reduced the volume of PCR initially here by 5-fold, but then we also wanted to test reducing the amount of input DNA. And then you'll see on the far right, these are echo calibrations that we have tested internally and are good to use for these reagents. So we know what calibrations need to be used to transfer these solutions. The other great thing about the Echo is you'll see in the protocol for the forward and reverse primers, the protocol recommends using a 1 micromolar concentration and high volume, whereas with the Echo, we can use master stock concentration with very low volume. So you'll see we're transferring 50 nanoliters of 100 micromolar primer, which is still an equivalent ratio to the protocol. Down below, we see our PCR cycling conditions and 25 cycles of amplification. Next, you'll see the second PCR, where the reduction is more drastic. Here, it's a 1 to 10-fold reduction. And another really great point is that I was able to purchase all of my sequencing adapters through IDT, and IDT was able to provide barcodes, sufficient barcodes for 384 multiplexing in an Echo qualified 384 polypropylene plate. So I ordered this from IDT. It came in an Echo plate ready to use. I did not have to do any further transfers. I was able to just transfer it directly from that plate, 50 nanoliters at a time. Once again, Echo calibrations on the right for transferring these solutions and our PCR cycling conditions below. So once that is all done, we wanted to QC our library, and here you'll see electropherograms of each of the samples that we tested. So in the Microbiome sample, it's 8 bacterial strains and 2 fungal strains, and you'll see that we are getting our expected 450 base pair peak with a little bit of nonspecific binding around 650. In the ATCC community panel, that was 20 strains of bacterial, and you'll see once again that 450 peak with just the slightest bit of nonspecific binding. In our negative control, you'll see the absence of that 450 peak, although you do see some other nonspecific amplicons that are being created. Whereas in our positive control, Staphylococcus aureus, you'll see a very strong and sharp 450 base pair peak, which is what we expect. So a lot of this data was then taken and ran through CosmosID. And here you'll see that in this chart, on the left we have the organisms. The gDNA abundance percentage is what is given to us from the manufacturer for these mock communities. And on the right, you'll see the percentage of reads aligned to each organism over the total. So these are all percentages, and from decreasing amounts of input, from 2.5 nanograms of input all the way down to half a nanogram of input. And you'll see that as we decrease the amount of input DNA, that these values do not change significantly. So the answer stays the same regardless. And you'll notice the two fungal strains on the bottom, Saccharomyces cerevisiae and Cryptococcus neoformans, they're fungal, and so the 16S rRNA amplicon did not pick up anything for that. At the top, you'll see Saccharomyces cerevisiae is producing very, very low reads with the slightest amount of bacterial contamination. And on top, Staphylococcus aureus is providing us with very strong 95% reads are mapping to Staph aureus. Okay, moving on to the next slide. Here we have lots of data from ATCC. These are the 20 strains that are available. And same setup, you'll see the organisms and the gDNA abundance is what is given to us from the manufacturer. And on the right-hand side, we've tested our 1 to 10 miniaturized reactions with decreasing amounts of input DNA. And once again, you'll see the trend stays the same across different input amounts from 2.5 to 1 to half a nanogram. The answer, the read percentage continues to be consistent across all three. So that gives us confidence to not only miniaturize the reaction, but also in cases where we have very little sample input, be confident that the answer would be the same. So here I'm showing just a little bit of CosmosID data. I hope to not play spoiler too much, but you'll see that on the left, the bottom left, the alpha diversity measurements, what we are looking for is a tight triplicate clustering of samples. So samples 1, 2, 3, there are zymal replicates of decreasing amount of input and so forth. You'll see that this triplicate pattern is very consistent except for when we reach Staph aureus, where the diversity is so low that any little bit of noise can throw those points off. However, on Saccharomyces cerevisiae, we see the diversity stay very, very low and very consistently. On the right-hand side, this is an important graph. The rarefaction curves generated by CosmosID, this helps us determine how many reads are necessary in order to saturate the diversity of the sample. So the very bottom cluster you'll see is the zymo sample, and you'll see that they all give the same answer and saturate the sample diversity well before 100,000 reads. The middle cluster that you see is ATCC, and once again they cluster well and they saturate diversity in comparison to reads at about the same point. There are two outliers on top. Those were manual controls that were just extra abundant in terms of reads, and so that's why you see that there was so much more there. Okay, here we have a Bray-Curtis distance graph based on similarity. So you'll see that similar samples, all the zymo replicates cluster together with strong connections. You'll see that the ATCC as well as the Saccharomyces cerevisiae all cluster closely together. Whereas there are other samples, the Staph aureus, they were a little more noisy. You'll see them spread out a little further. So this is just an example of what CosmosID can provide to you as far as the data visualization and analysis go. Okay, moving on to miniaturized whole genome sequencing. I just wanted to show the workflow once again. We have our microbial gDNA harvested and purified. Then we go through tagmentation, 5 minutes, 55 degrees, and then an indexing amplification, which we do 14 cycles of amplification there. Library QC, fragment analysis, quantitation, then we're able to normalize and pool all of our libraries together very rapidly in the same step using the 25 nanoliter transfer increment. Then we're able to denature these libraries and load them onto the sequencer. For this experiment, we chose to analyze the data in two main ways. One is a very old school, low throughput, traditional alignments to the reference genome using Biomatter's Genius software, and the other was to upload our raw data to the CosmosID Metagenomics platform. And I just have to say that the CosmosID Metagenomics platform was able to quickly analyze all of our data, all the replicates, in 30 minutes, whereas, as many of you know, manual alignments to the reference of each replicate can be very compute intensive, especially if the user does not have the proper equipment. So here is a little more detail on the workflow. We tested 3 samples. ATCC, NIST had a challenge sample that was done in collaboration with CosmosID, as well as the Zymo microbial standards. These samples were at 1 nanogram per microliter, and we performed quadruplicates. And so you'll see here that we've done a 1 to 10 reduction in volume size, keeping the ratios the same. And on the far right, you'll see that the Echo is able to transfer these solutions, and we have these calibrations for the user that have been validated in-house. Moving on, you'll see the conditions and details for the indexing amplification. Once again, you'll see that our indexing primers were reduced 100-fold in volume, and that is just due to the fact that we are able to use our indexing primers from IDT and the plate at 100 micromolar concentration. Being able to accurately transfer 50 nanoliters is definitely a plus here. So we did a 14x cycling for our amplification, and then SPRI bead cleanup as follows below. Now, magnetic beads can be an expensive part of the workflow as well, and so being able to use much less of those due to a reduced volume reaction, is also providing savings for the amount of beads that are used. So after this was done, we did a picogreen quantitation of these samples. You'll see at the top, the standard curve was generated using the Echo. For that, I used the standard lambda DNA and only had to provide 1 concentration of that, and the Echo was able to precisely dispense the amount of DNA necessary for each of these standard points. And so there is no serial dilution involved in this. I simply just asked the Echo to transfer to the amount of total DNA to hit each of these points. And so you'll see at the bottom that our yield, despite a miniaturized reaction, the yield was still very good. We were able to get about an average total yield of 8.19 nanograms per microliter, and the CV is about 8.5%. Moving on, I'm just showing some electropherograms here from the Agilent tape station that we use in-house. You'll see that all 3 samples produced very similar fragment size profiles, and so I was confident to go ahead and assume they were all similar in my normalization calculations. You'll see that there's a long right tail, which is also nice for our chosen run length, which is 2 by 300s on the MySeq. We're using a V3 kit that's capable of 600 cycles total. So 2 by 300 gives us nice big reads for these genomic assemblies. So taking this data and moving it into CosmosID, here you'll see on this chart that on the left-hand side, the reference is what is given by the manufacturer, and for CosmosID, we were able to average all 4 replicates in, as well as the CVs for those. What you'll see in the total matches column is how much of the reference genome is being covered by the reads that we generated, as well as the CV of that. On the far right, you'll see the Genius alignments that were run on my laptop. These took a long time because I did not have a computer that was built for this sort of heavy computation. You'll see that our average abundances fall pretty closely in line with the reference, given the enzymatic process and some of the biases that could have been introduced from that. Moving on, the ATCC sample, you'll see that this chart is set up in a very similar way, where the reference is referring to the amount of gDNA abundance per weight. And so our average abundance in general, again, is pretty close to what the manufacturer is specifying. The CVs are also pretty low, with the exception of a couple. And the total matches, you'll see that the genomes are decently well covered, but we could have went with higher reads potentially to cover more of the genome. The total matches CVs are all pretty good. They're below 6%, and so we're confident from replicate to replicate that we're producing high quality and similar answers, and that gives the customer hopefully some confidence as well that they do not need to replicate their experiments so much. Genius alignments, again on the far right, was performed for one of the 4 replicates, and this took my computer all day. But you can see that all the answers are pretty similar. They all tend to agree, and so we can trust that miniaturization has little effect on these numbers here. Lastly, here is the NIST sample that was generated in collaboration with CosmosID. This was a sample in which we did not know what the organisms were within, and so we were not able to run genius alignments to the reference because we didn't know what the reference was. So CosmosID was able to rapidly align these and find, once again, average abundances over 4 replicates as well as the CVs. You'll see that the answer here is extremely tight with the exception of E. coli, which is still under a 10% CV across 4 replicates. And that you'll see that the total matches are all pretty good. The genome's pretty well covered, and the CVs of these replicates are very tight. Okay, and moving on, we have a visualization produced by CosmosID. Here you'll see all 4 replicates of these samples put into a stacked bar graph, log scale, and normalized to 100%. You'll see the answer across all 4 replicates are very consistent. There's very little change here. And that gives us confidence that it's a very consistent process across, and we do not need to use this many replicates. After that, we've also been able to take a principal component analysis graph from CosmosID. So you'll see that each of these samples color differently. They all cluster closely together, showing that they have high degrees of similarity, though they are spatially separated from each other. And this is what we want to see in a principal component analysis. So with that, I'm going to summarize our findings. 16S rRNA Amplicon sequencing, that can be miniaturized effectively. And we find that using the rarefaction curve, that we were able to not lose any samples, and diversity was maintained. There's no loss in data quality. So that can be confidently miniaturized, but always use the rarefaction curve as a guide for how many reads are necessary in order to saturate your community sample. Whole genome sequencing can also be effectively miniaturized with no loss in data. That we see that the diversity is maintained. We did not lose any strains when going through the CosmosID analysis. And lastly, I'm hoping that this cost-reduced miniaturized whole genome sequencing will open the doors and broaden the use of the technique in hopes that one day it will become such a ubiquitous and routine process that it's used everywhere. And with that, I would like to say thank you, and I would like to turn it over to Dr. Hasan from CosmosID, who will tell you much, much more about the back-end bioinformatics abilities of their very powerful platform. So thanks again, and take questions at the end. Appreciate it.
Thanks, Jefferson. And now over to Nur.
Thank you, Jefferson. Hello, everyone. Welcome to the second part of today's webcast. My name is Nur Hasan. I am the Chief Scientific Officer at CosmosID, the leading microbiome solution company located at Rockville, Maryland. In this part of the presentation, I'm going to introduce CosmosID bioinformatics platform to you and describe how it can be used for large-scale data analysis for cross-disciplinary microbiome research. However, before describing the platform, I would like to discuss briefly about the growing importance of microbiome and what challenges we are facing today to translate microbiome findings into product development. Subsequently, I would also like to offer a demonstrations of our platform. As human and microbiomes have co-evolved together, complex interactions between them are becoming a critical part in our health and well-being. Like as our healthy microbiome directly benefit us by regulating our innate immune system, contributing to the biosynthesis of essential vitamins, metabolizing lipids and carbohydrate for us, and creating a protective barrier against pathogen colonizations. Imbalance to these healthy microbiome have also been implicated with a broad range of disease states. In this slide, you can see a list of wide spectrum of diseases where residence microbiome and/or its dysbiosis has been implicated, ranging from neurological, oral, skin, respiratory, and intestinal diseases to autoimmune or even metabolic disorders. Interestingly, this list is continuously increasing, and we see a steady cast of reports every now and then demonstrating yet another association of microbiome with another health conditions. Such growing understanding of residence microbiome and their association with variety of health conditions provides us enormous opportunity to develop novel diagnostics or therapeutic approaches. Therefore, no wonder why microbiome is becoming the target for new product development. So far, shotgun DNA sequencing has been the dominant technology for determining compositional and functional aspect of the microbiome, and it was reasonably successful in linking microbiome dysbiosis to numerous diseases as we discussed in the previous slides. However, discovery of candidate molecules and pathways that underlie mechanism still remains as a major challenge to us. And to address those challenges, we actually need a system-level understanding. In addition to microbial community profiling, we do need to explore secretomes and metabolomes of the community to understand how microbiome affects various host responses. Similarly, we also need to measure the numerous ways hosts are responding to their residence microflora and identify novel proteins and pathways mediating such interactions so that we could delineate cause and effect and identify druggable targets. Apart from these, we also experience additional challenges throughout the entire microbiome sequencing and analysis workflow. For example, microbiome study often does not have statistically significant sample size. Sometimes that's because of increased cost of sequencing. And to this end, the miniaturized, cost-effective method that Jefferson presented earlier today would be a perfect way to accommodate large-scale, statistically significant sample size into microbiome study at affordable cost. We also observed lack of controls and measurement standards in many microbiome studies, contributing to biases and reproducibility issues in their study outcome. In the bioinformatics part, key issue has been sensitivity and accuracy of tools being used and their inability to classify organism at subspecies or strain level. Now the question is why you need strain level identification in microbiome study, an attribute that most of the existing bioinformatic tools fail to provide. The short answer is the clinically informative and actionable unit in microbiology is actually a strain, not a species. To demonstrate that, here I am citing an example of a classical probiotic species, Lactobacillus casei. As you can see, 3 distinct strains of Lactobacillus casei are used for 3 very distinct applications, all related to fermentation of foods. Strain 1 is used here for ripening cheddar cheese, whereas strain 2 has been used for fermentation of Chardonnay wine. A third strain is used as an immunostimulant, like probiotics in Dan Active. So 3 distinct strains of a single species conferred 3 very different biological consequences, indicating that strain identification is very critical for microbiome study. Now let's discuss the challenges we are facing today in data analysis part. The most critical factor in microbiome analysis is the database, as your analysis is as good as the database you use. Unfortunately, most of the tool uses only bacterial database, but it is absolutely critical that database represents all kingdoms, not bacteria only. It must have broad and deep representations of non-bacterial microbiota like archaea, fungi, protists, viruses. It is also critical to make sure that database have adequate representation of commensals and environmental microbes in addition to pathogens. We need to ensure that databases are well maintained, version controlled, and periodically updated. In the algorithmic side, the critical point is to ensure the tool provides necessary sensitivity and specificity of detections and actually able to precisely identify organisms at subspecies or strain level, for the very reasons we discussed in the previous slide. It is also important to understand how much computational time and cost it incurs, whether the algorithms are professionally maintained or not, as most of the freely available open source tools are not well maintained or updated after the publication. But these are critical for backward and forward compatibility of your result. So keeping all this consideration in mind, we developed a comprehensive microbiome bioinformatics platform as well as a robust end-to-end wet lab workflow to offer a one-stop solution for microbiome scientists. Our state-of-the-art NGS service laboratory featured with high-quality, bias-free sequencing services offering expert consultation on study design, sample prep, collection, preservation, to all other downstream processes. For bioinformatics, we developed an user-friendly cloud bioinformatics platform which utilizes ultra-fast bioinformatics algorithm and expert-curated genome database, offering multi-kingdom microbiome analysis at subspecies and strain level. It also offers ability to simultaneously profile community resistance and virulum and perform real-time comparative metagenomics and statistical analysis on the fly. In a nutshell, the platform provides the scalability, precision, speed, and resolution needed for any large-scale microbiome studies. To this end, we built the world's largest and most comprehensive database comprising over 150,000 genome and gene sequences and representing both commensals, pathogens, and diverse environmental microbes. Besides the curated content, another most important feature of our database is it is organized as a phylogenetic tree. Our team has invested years in curating this database, the content of which is then used to build phylogenetic trees like virtual tree of life. Algorithm is then go and find hundreds of millions of biomarkers that are uniquely identified across every branch, node, and leaf of the tree. Such phylogenetic organizations of the database enables us to identify and differentially detect closely related species and strains as they often represent by distinct nodes or branch of the tree with uniquely identifiable biomarkers. Subsequently, we employed machine learning techniques to develop filter threshold by analyzing thousands of in silico laboratory-generated and orthogonally validated datasets comprising varying level of microbial diversity and complexity, and helped achieve significantly increased detection specificity. There is always a trade-off between sensitivity and specificity, and most algorithms achieve robust sensitivity by trading off specificity, effectively accepting false positive calls. Here we are using a phylogenetically structured, curated database to achieve high detection sensitivity and precision, followed by machine learning filter algorithms to secure high detection specificity. A balanced combination of these two approaches enabled us to deliver industry-leading specificity without sacrificing the detection sensitivity. And you will evidence this in next few slides. At CosmosID, we are continuously evaluating the performance of our platform both through in-house evaluation as well as independent validation by third parties. Here are some results that confirm the industry-leading performance of our solution. This slide shows the results of an internal benchmarking study using combination of over 30 in silico and biological datasets and following the method proposed by a team of Cornell researchers. As you can see, the species level result at the upper left graph demonstrates CosmosID performing favorably in both precision and recall compared to any other tools being tested. It also demonstrated that there is very little trade-off between precision and recall, as evident by the highest F1 score. Looking at the bottom left figure in the slide, you will see that the performance at subspecies level is even more dramatic. Where all other tools significantly fall short of performance at subspecies level, CosmosID was able to substantially retain both its precision and recall. The next figure in the middle of the slide represents a recent challenge called Mosaic Clinical Stain Detection Challenge 1, sponsored by Johnson Human Microbiome Institute and DNA Nexus. The focus of the challenge was to assess the ability of bioinformatic tool for strain level identification and abundance prediction. We are very proud that CosmosID won this challenge by delivering microbiome analysis at strain level accuracy and industry-leading sensitivity and precision. Additionally, our performance on the biological sample was 64% higher than the score of the second best submission. Furthermore, we also have been awarded with 2018 Water Technology Idol Award for our seminal work on metagenomic-based pathogen detection system for water safety and reuse. In this slide, we represent the results from another recent competitive challenge organized by Precision FDA Center for Food Safety and Applied Nutrition. Here also, CosmosID became the leading tool for strain-level pathogen detection with lowest false positives at all different sample categories. We essentially correctly identified the highest number of culture-negative samples and outperformed all other contenders in strain-level accuracy. Now I would like to perform a live demonstration of our platform. And to do so, using a browser, I'm navigating to app.cosmosid.com. Once you arrive to the landing page, you can register for a new account. But if you already have an account with us, just use your email ID and password to log in. Once logged in, you can explore the navigation panel on the upper left side. At the bottom of the navigation panel, you will find information related to your account, like how many analysis credits you have currently, and option to buy additional credits using our e-commerce portal. To start the analysis, first we need to upload the data to our platform and select appropriate sample type. If you like to analyze a shotgun dataset, select Shotgun Metagenomics here. And then simply drag and drop your datasets right here. Or click Open the File Browser and select the datasets of interest. Additionally, you can also import datasets from your Illumina BaseSpace account by using Illumina BaseSpace import option available here. Another way to bring data to our platform is using our command line API available at GitHub. You can explore our documentation page for further details about the data upload options, user guide, and other relevant information. Once a sample is uploaded, analysis will automatically start and results will be populated in the dataset folder. Under dataset folder, you can keep your datasets organized project by project using folder structure. And in the upper right corner of the panel, you have option to add new folder. Now let's explore some results from healthy microbiome sample derived from human microbiome project. Under subsample healthy HMP folder, I have already put some representative samples. Let's select a tongue sample from here. Panel on the top shows some statistics about this particular dataset. As you can see, it shows how many organisms were detected, number of reads hit to the database, and total read depth. By clicking to the eye, you can open up metadata window for annotation. You can fill up appropriate fields and update this. We have few different options of results. So let's start with table view. By default, table view shows all different strains and strain level statistics. But using the taxonomy switcher at the top, you can switch to other taxonomic level if you like. It provides a list of strain identified in the sample, also shows their corresponding tax ID. If you click to the tax ID, it will take you to NCBI taxonomy browser. You can also see statistics on frequency, percent of unique match, which is the number of different chimeras found in the sample that are unique to a specific strain divided by the number of pre-calculated total possible unique chimeras present in the reference database. Percent total match, which closely surrogates physical coverage of a genome, and relative abundance, which is normalized based on the genome size. You can also review the documentation page for more information on these statistics. Now we can apply the machine learning filter threshold that we have discussed earlier to improve detection threshold. By applying the filter, you see the total number of strains detected reduced from 452 to 316 highly confident calls. Now let's explore some visualization tool we have here. First, we can visualize the data using Chrona. Here you can zoom in to a particular taxa of your interest and zoom out as necessary. We also have bubble graph, where size of the bubble is proportional to its abundance. We have a collapsible tree, and you can actually trim the tree at any taxonomic level by selecting the appropriate one from the taxonomic switcher. We also have radial tree, which is one of my favorite, and you can switch different taxonomic level or even change the filter threshold from total to filter, and the visualization will be automatically updated. Additionally, we also have donut chart. Here you can choose either frequency or relative abundance, and also you can adjust the tail if you like. We also have stacked bar graph with similar functionality. As you see, all the visualizations are very interactive and exportable to PNG or other vector graphics. You can also export the matrix in CSV file or enter results in PDF format. As this was a shotgun metagenomic sample, therefore we have opportunity to explore results from other kingdom of databases. So let's explore results from other databases. First, we have to select antibiotic resistant database. Looks like these samples contains few beta-lactamase, tetracycline, macrolide, and aminoglycoside resistance gene. Now let's move to the fungi database. The result shows no fungi detected in this sample. Next, to protease database. No protease detected in these datasets. Now viruses. We see several phases detected, as our viral database also included phase. Next, we can look at the virulence factor database. As you can see, many different virulence associated genes are detected. Now that we got a multi-kingdom view of metagenomic analysis, let's explore what comparative analysis we can do in this platform. These are different comparative analyses I have done in the past, and this can be reviewed anytime when you like. But let's start with a new comparative analysis. And for that, we have to click the Create button on the upper right corner. First, we have to give it a name, say Nature Demo. Then we have to select a database, let's say like bacteria. It takes a moment to load all the data. Now we can select filter sets. We can select either total or filtered. And for attributes, we can select like relative abundance, frequency, percent unique match, or total match. Now we have to annotate the cohort levels. We have up to 24 cohorts option. For this demo, I just need 2. So let's say we use C for control in level 1 and D for defective in level 2. Now we can search samples by name and assign them according to their right cohort. Once done, we can submit it. It will take like a minute to complete the analysis. So it creates a matrix table at species level, and you can sort based on the cohort or by individual sample. You can change the view to other taxonomic level using taxonomic switcher at the top, like phylum level. You can make heat map and assign the tail, let's say 5%. If you hit apply, lowest 5% will be categorized as others. You can get a stacked bar graph as well, same as before. You can annotate the tail if you like. You can also use random color. As you see, there is a continuous presence of Deinococcus thermus phylum present in all defective cheese, but it is not present in control, which indicates that it might be the possible cause of pink discoloration in cheese. Next, we can see 3D PCA based on covariance. As you can see, control and defective samples are clustered separately. We also have alpha diversity here, and in alpha diversity we have option for Chow-1, Shannon, and Simpson. In all cases, defective cheese have higher alpha diversity than control. Next, we go to beta diversity PCOA. Here we have PCOA based on both Jacquard and Bray-Curtis. We also have beta diversity network graph and read statistics. Again, remember, as you see, all of the visualizations are very interactive and exportable to PNG or vector graphic. You can also export the matrix in CSV file and enter results in PDF format. So that's the end of my today's demo. Let's go back to the original presentation deck. So what we built here is a cloud interface that is simple but precise, user-friendly but comprehensive, and powerful. It is very secure and scalable, therefore you can reliably use it for large-scale microbiome analysis. In addition to CosmosID web app, you can also access it through command line API available at GitHub. It is also available at Illumina BaseSpace and Thermo Fisher Cloud. And a plugin is also available through QIAGEN CLC Genomic Workbench. End of the day, we focus on the entire microbiome workflow, providing high-quality, bias-free sample prep to sequencing service, along with high-resolution, ultra-fast bioinformatics. Our aim is to enable microbiome scientists to accommodate increased sample size at affordable cost and carry out ultra-fast bioinformatics with high accuracy and precision needed for translational outcome. This slide describes the major areas we are currently working on. where significant microbiome discoveries and product developments are apparent. And here you see a short list of our clients, collaborators, and valuable partners out of several hundreds we are working on. Here is a special announcement for you guys today. Receive double free credits today if you register a new account at app.cosmosid.com. Finally, feel free to contact us and give us a free trial and also take advantage of our special offer of $199 shotgun sequencing. And with that, I would thank everyone for attending this webcast.
Thank you for your presentation, Nur and Jefferson. It is now time for the Q&A. To ask the speakers a question, just type it in where it says type your question here and then press submit. So our first question, and this one is for you, Nur, and it asks, can your platform analyze data sets from other nanopore sequences?
Thanks for the nice question. Answer is yes. You can analyze any sort of high error long read sequencing, like both from Oxford Nanopore or PacBio sequencer using the CosmosID platform.
Perfect. Thanks, Nur. And our next question asks, do you happen to see sample dropout from the reduction of library volume size when profiling 16S samples? Is that one you can answer, Jefferson?
Yeah, sure. So we in the samples that we had, we did not see any sample or strain dropout from the reduction of library volume size, especially with the 16S process that is highly amplification based. Even at half a nanogram for our samples, that was plenty of DNA. We weren't hitting the limits of complexity there. Obviously, if you miniaturize further, it's possible to lose samples, but we would like to emphasize using the rarefaction curve as a guide to figuring out how much.
Perfect. Thanks, Jefferson. And our next question is for you, Nur, and it asks, you demonstrated in your presentation that strain resolution is important. Can you please explain why you perform better than others in that specific aspect?
Excellent question. Actually, the reason for this improved performance and strain level resolution relies on three distinct elements that is unique to our database and the platform. First, our database is expertly curated and the content has been organized as a phylogenetic tree. Along with those phylogenetic organizations and curated content, we also use a machine learning techniques that helps improving specificity of detection by reducing background noise from signal.
Excellent. Thanks, Nur. And our next question asks, can you also explain how you actually pull the samples and what is your final volume? Jefferson, is that something you can answer?
Yeah, so after we create our libraries, we get some sort of a feedback, QC feedback, so that would be fragment size analysis, as well as some sort of quantitation, which can be fluorescence-based or qPCR-based. Our latest favorite has been to run a cheap and small sequencing run. For example, a MySeq Nano, or on an iSeq, you can do just a run just to get the index distributions. Once you get the quantitation from qPCR, DNA quant, or a small indexing run, then you can normalize your samples based on how much your highest sample and how much of each additional sample needs to be added in order to reach that total amount. We build that work list, we put it into the Echo, and then they pool it into one or several wells depending on your destination labware. So for a 384 well plate with 384 unique samples, the pooling volume we tend to set around 200 nanoliters, and the whole plate can end up being about 50 microliters. Yeah.
Excellent. Thanks, Jefferson. And our next question asks, what is the advantage of organizing database as a phylogenetic trees as opposed to a taxonomy-based organization? Hasan?
Another very excellent question. Traditionally, database has been organized as a taxonomically. However, the uniqueness of phylogenetic organization provide you opportunity to achieve subspecies and or strain level identification. In a taxonomic scheme, you get at the lower level species and then directly to strain. However, if you organize a database in a phylogenetic tree, like as a tree of life, you will see that phylogenetic tree often offers specific clade or branch for each of the subspecies, serovar, pathovar, and biovar, thereby allowing you classification at a much finer level to achieve strain or subspecies level identification. Secondly, there is also another benefit that we always experience, that the natural diversity of strain is great. So we often encounter novel strain or species in a query data set. So by organizing phylogenetically the database, it helps us to identify the organism, even if it is not a novel strain that is not represented in database, we will be able to place that to its phylogenetic near neighbor level. And third reason is we use genome sequencing data, which is genomic data, and probing them against a genomic database. Therefore, it makes much more sense to organize the database as phylogenetic tree so that the data can be right way probed to each other. On other hand, in taxonomy use phenotypic data, and we are not classifying phenotypes here. We are actually classifying genotypes. Therefore, it makes sense to organize that database in a phylogenetic tree, and that definitely help us in precision in identification process.
Excellent. Thanks, Nur. And our next question asks, when transferring your primer at a higher concentration, do you compensate for dead volume? And how often do you typically go back to the same source plate? Jefferson?
Yeah, so when we order our plates from IDT, the sequencing indexing primers are delivered at 100 micromolar concentration, and the wells are filled to a maximum volume of 60 microliters. So if we assume about 20 microliters, and that's a safe bet for dead volume, then we have about 40 microliters of working volume. And so if we transfer 50 nanoliters at a time, 40 microliters, 50 nanoliters, you could, in theory, transfer from that plate 800 times. And so potentially it'd be good to use this plate for 3 months or 6 months, depending on the throughput of the lab. And then you should be pretty safe with the amount of liquid there, because 50 nanoliters in a 60 microliter well is very little. So you'll be able to transfer many times out of that. And then as far as the contamination question goes, we've done qPCR studies where we transfer into one well and leave the surrounding wells open, and we don't see any transfer contaminations. And those are available on the Labcyte website. Thanks.
Excellent. Thanks, Jefferson. Well, I'm afraid that is all we have time for today. I would now like to thank Jefferson Lai and Dr. Nur Hasan for their presentations and for answering your questions. I would also like to thank the webcast sponsors Labcyte and, of course, you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.